Good morning, everyone. In this section, I'll be talking about the contribution of another eminent scientist under your syllabus, that is in section A of Unit 2, CCF Microbiology Semester 1, that is Carl Woese. Carl Woese, three domain of life. Carl Woese, three domains of life. Okay. So, what are the three domains? First one is bacteria. Third one is archaea or archaea basically. And last but not the least, that is eukarya. Okay, so these are the three domains of life: bacteria, archaea, eukarya. Bacteria, as you all know, it's obviously bacteria, unicellular, simple life forms. Archaea means the archaea. Eukarya means the eukaryotic organisms like us, or it can be fungi, it can be plants. Okay, so three domains of life. And this classification of the Carl Woese basically depends on two factors. One is the evolutionary relationship, and second is sequences of ribosomal 16S RNA. Basically 16S rRNA sequences and the evolutionary relationship, because these two factors are very it basically they are the very linked terms or linked topics or parameters. So Carl Woese three domains of life actually based on the molecular characteristics of the life form. Okay, in the previous video we have already learned about the Whitaker's 5 kingdom classification, which is based on the cell structure, their complexity, food nutrition, how they survive on that. But in case of Carl Woese three domains of life, he actually focused on the molecular part, which is more important than any other kind of a characteristics. Molecular part, only the molecular basis you can identify any kind of a microbes or any kind of organism specifically. Okay, morphological you can identify, but 100% assurance will be given by the molecular studies only. So molecular studies mean, as you all know, that if you want to identify any kind of a bacteria, in case of microbiology, if you identify any kind of a bacteria, you have to first extract that whole genome, not the whole genome but the genomic DNA. Okay, you have to extract that genomic DNA with the kit and all, genomic DNA isolation kit. Then you have to amplify that specific gene. So in that case 16S rRNA. So you have to amplify 16S rRNA gene with the help of polymerase chain reaction, and then you have to sequence it, and you can easily identify what is the bacteria, the unknown bacteria. Okay, from that you can also get the evolutionary relationship, means in a simple form, who is the father and mother, who is the brothers, who are the sisters, who are the uncle aunties of that bacteria. As we have the families, bacteria or any kind of organism also have families. That is the evolutionary relationship. Okay, so if you want to identify really the parents or the sisters, brothers or the grandparents of that bacteria, you have to first sequence the 16S rRNA gene of that particular organism, and then you have to make a phylogenetic tree. Okay, phylogenetic tree. This is very important term. Phylogenetic tree you have to make, and from that you got the evolutionary relationship of that unknown particular bacteria, which have identified with the help of 16S rRNA gene sequence. So this classification is basically depends upon the molecular characterization of any kind of life forms. Okay, so what is the utility of this classification? Obviously you can get a clear idea about upgraded phylogenetic tree. These are very important topics, and I know it's very much not known to you guys. later in your syllabuses, in your semester, you will learn about how to make phylogenetic trees and all. It's a very important topic. You will loved it when you learn how to draw a phylogenetic tree, or with the help of the softwares, how you will make the phylogenetic tree of the unknown bacteria after identifying them with the help of 16s RNA gene sequences. In my video, I'll also teach you with the help of the softwares. So for that, you have to subscribe my channel. Okay, jokes apart. Now you will know about the phylogenetic tree making, evolutionary relationships, okay. So these type of things you will learn about from this Carl Woese 3 domains of life. So with the help of these 3 classifications, means the binomial nomenclature, I have already made videos on this and also on Whitaker's 5 kingdom classification. And now today, Carl Woese 3 domains of life, that whole diversity of microbial origin or any kind of a life form, you will learn about it. And that is basically the part of your section A under UG SEM 1 syllabus. Just one topic on which I'll make video, that is the differentiation, how we'll make differentiation between the eukaryotic and prokaryotic organisms, or even also in case of the microbes. I'll make the video and upload it in my channel. You guys, remember thing, you guys have to follow your notes, your textbooks as suggested by your colleges. But if you really want to learn and in a point wise manner, in a very short term, you can watch my channel, because I'll definitely try to make things easier, not in a very elaborate part, because I don't also loved it. That is why I tried to make it in a point wise manner, just to make you guys understand it, as it's a very new topic to all of you. So thank you so much. If you like, please subscribe my channel and share with your friends. Bye.